Hello everyone, Mitch Albers here. In this biocast, what we're going to do is finish up our discussions on cellular respiration, where we're going to look at the overall ATP energy production. We'll talk a little bit about how life avoids equilibrium and talk about some of the diversity of cellular respiration in terms of some of the metabolic pathways that feed into cell respiration and how it's regulated. So let's get started and look at the overall ATP production. So first of all, let's look at the direct ATP energy output from cell respiration. You recall back in glycolysis, we produced 2 ATPs directly out of glycolysis. In addition, we produced 2 ATPs directly out of the citric acid cycle, or the Krebs cycle. Okay, and remember that those 2 ATPs produced in glycolysis and the 2 ATPs produced in the Krebs cycle were produced by substrate level phosphorylation. The third phosphate that was donated onto the ADP molecules came from one of the substrate intermediate molecules in the glycolytic pathway and in the Krebs cycle pathways. The ATPs that got produced out at the electron transport system, or oxidative phosphorylation, those ATPs are produced through oxidative phosphorylation. Okay, so in the end of our last discussion on the electron transport, we mentioned that the energy equivalencies for the electron carriers, NADH and the FADH2s, that the energy output is about 2.5 ATPs for every NADH that goes into the electron transport system, and for every FADH2 we get about 1.5 ATPs. Okay, now I also mentioned that we're going to round those numbers up just to kind of keep this a little bit, the accounting a little bit easier. So we're going to just say that for every NADH we're going to get 3 ATPs, and for every FADH2 we're going to get 2 ATPs. Okay, and that'll kind of help us come up with our final tally for the energy production here. Notice that there's a range of ATP production here. That if we looked at what is the potential energy of a glucose molecule, if everything was working as efficiently as possible, we could probably get 38 ATPs out of it. But due to some of the constraints of how the cells manufacture those ATPs, the conditions that they operate under, we're never going to achieve that potential. And then we also have to abide by the laws of thermodynamics on top of that. So we're going to use our energy conversion of 3 ATPs for every NADH, 2 ATPs for every FADH2 to do our accounting here. So let's start over on the far side here. Remember that out of the Krebs cycle we got 6 NADHs and 2 FADH2s, which then went into electron transport. Okay, so if we said that for every NADH it was equal to 3 ATPs, okay, so this would be 6 times 3, which would be 18 ATPs just for those NADHs that came out of the Krebs cycle. The two FADH2s, since they're worth 2 ATPs each, that would be 2 times 2, which would equal 4 ATPs. For those, just these two FADH2s that come out of the Krebs cycle. Okay? We get 2 NADHs that come out of pyruvate oxidation, so that would be 2 times 3, which would equal 6 ATPs. And then we have to deal with the 2 NADHs that come out of glycolysis. Okay, and remember, this is happening out in the cytoplasm, and there's a bit of an issue of getting those NADHs into the mitochondria and to feed into the electron transport system. Okay, and so the electrons that are picked up from glycolysis, okay, and eventually are going to get delivered to the electron transport system, have to go through those mitochondrial membranes through shuttles. Okay, and so there's two possibilities here what might happen, and that is that the 2 NADHs that get produced can convert those electrons and get picked up by 2 FADH2s. And if they do, remember, if every FADH2 is equal to 2 ATPs, then this would be 2 times 2, which would equal 4 ATPs. Okay? And then for the 2 NADHs, you know, for these shuttles to work, we could say that it also costs the cell 2 ATPs just to shuttle those electrons to be picked up by NADH within the mitochondrion that then can go to the electron transport system. So it's a bit of work getting those electrons out in the cytoplasm into the mitochondrion for ATP production. So when we look at it, it's going to cost the cell a couple ATPs for doing that work. Okay, and we'll take that into account here when we add everything up. Okay, so so if we do the addition here, we've got the 18 ATPs from the 6 NADHs from the Krebs cycle. We have the 4 ATPs from the 2 FADH2s that came out of the Krebs cycle. We have the 6 ATPs that came out of pyruvate oxidation, from the 2 NADHs that came out of pyruvate oxidation. Okay, and then let's just say we're going to use the 2 FADH2s that came out of glycolysis, where the electrons were shuttled from NADH to FADH2s. So that was a conversion of 4 ATPs. We do the math, 12, 18. We end up with 32 ATPs total. Okay? Add up those 32 ATPs plus the 2 ATPs from glycolysis plus the 2 ATPs from the Krebs cycle, and we see that we can get about a net of 36 ATPs overall from a single glucose molecule. Okay? Now, again, there's a range of how many ATPs will actually get produced, and again, it'll depend on the type of cell, the conditions that cell's operating under, but just to conceptualize, that's kind of how we can kind of determine, you know, how the yield in ATP energy occurs during oxidative phosphorylation, and how we sum everything up. We're acting as the bean counters here, the accountants for cellular respiration, and looking at the overall energy production. Now let's think about another concept here. Kind of going back to our discussion on the electron transport system, we talked about the role of oxygen. remember, oxygen's role is to pull electrons off the end of the electron transport chain. They combine with a couple of protons that get pulled out of the matrix, and they form water. Okay, so we constantly have to keep breathing in oxygen and delivering oxygen to our cells so that they can constantly be pulling electrons off the end of the electron transport chain. Imagine if oxygen wasn't available. Well, what would end up happening is these electrons would back up. And if there's no flow of electrons, okay, what ends up happening is when this whole system starts to back up, because there's no oxygen, then that ceases the pumping of these complexes in driving the proton motive force. And if we stop the pumping of these hydrogen ions into the intermembrane space, they will simply diffuse back to the matrix side, and they will do so until they reach an equilibrium. Because if the protein complexes that are responsible for pumping those hydrogen protons cease, we won't be establishing a concentration or electrostatic gradient. We'll lose our high energy potential, and when the hydrogen ions then diffuse back through the ATP synthase channel back to the matrix side, we could end up getting at some equilibrium point. That's when our delta G, our free energy situation, equals 0, that's equilibrium. And for life and cells, we want to avoid that at all possibilities. We never want to reach that equilibrium point. And that is why you constantly are eating foods in your diet. You're constantly breathing in oxygen and delivering oxygen to the electron transport system, so that we continually allow this flow of electrons, okay, down the electron transport chain. So that involves the constant pumping of hydrogen protons into that intermembrane space. That creates that proton motive force, which creates the high energy potential, so that there's a constant supply of a high concentration of hydrogen protons that then can diffuse back to the matrix side. So we have a constant supply of production of ATP, because we're going to need that ATP to do work. And we know that there's all kinds of work that needs to be done in the cells: protein synthesis, transport across membranes, catalyzing reactions, synthesizing products, all require an input of energy, and that work needs to be energized by ATP. So we want to ensure life evolve this process, that we're constantly avoiding reaching that equilibrium point. All right, so some of the versatility of cellular respiration is that, our focus here was just kind of looking at, how glucose enters the pathway, okay, starting back at glycolysis. And, but we should also understand that, in our diet, we take in complex sugars, we take in disaccharides, polysaccharide sugars, and through hydrolysis, through digestion, we break down those sugars, and obviously a lot of those sugars will get broken down into glucose, and it enters the glycolytic pathway. But we also take in other things in our diet. We take in fats in our diet, okay? And remember, fats compose of glycerol and fatty acids. Well, the glycerol, remember, that's a 3-carbon monomer group that goes into making up triglyceride molecules. Well, glyceraldehyde 3-phosphate also is a 3-carbon intermediate, and so the glycerol can get converted to glyceraldehyde 3-phosphate and enter the glycolytic pathway at this point, and then proceed with the rest of it. Likewise, the hydrocarbon chains, the fatty acids in fats can be broken down, and I'm sorry, those are CH2 groupings, right? CH2, CH2, the end would be a CH3. Well, those hydrocarbon, those CH2 groupings can be broken down into smaller molecules and can enter as 2-carbon molecules at the end of pyruvate oxidation as acetyl. Okay, and proteins can also be broken down into amino acids, and amino acids can enter at various points of the cycle. And one of the things that has to occur before any of those amino acids enter into the cellular respiratory pathway is that they remove the amino acid portion of the amino acid. So that's our NH3 grouping, and that is a process called deamination. Okay, ultimately, sugar is our ultimate energy source. When we burn up sugar, our second choice is going to be fats, and as a last resort, the third energy source would be proteins. Okay, but the first thing that our cells will metabolize are the sugars. So that just illustrates just a little bit of the versatility and diversity of how things can enter the cellular respiration process or pathway. And then the last thing we'll talk about here is just some basics on regulation. If we recall back in the metabolism chapter, we talked about metabolic pathways and feedback inhibition and how it's involved with regulating a metabolic pathway. Well, cellular respiration is a very complex metabolic pathway made up of sub-pathways like glycolysis, the Krebs cycle, and so forth. Okay, and one of the more key roles as far as enzymes in the overall process of cellular respiration is back in glycolysis. The phosphofructokinase enzyme acts as an allosteric enzyme, and it has various inhibitors and activators that play a regulatory role in keeping cellular respiration, whether it's upping its regulation or downgrading its regulation. Things like increasing the output of cellular respiration would be an increase in the amount of AMP. AMP is adenosine monophosphate. It's just like adenosine diphosphate or ATP, adenosine triphosphate. It's just the adenosine with a single phosphate attached to it. And when that concentration of AMP increases, that is a trigger that stimulates an activator. The AMP acts as an activator for phosphofructokinase to upregulate and increase cellular respiration output. The two indicating the diagram here, there are two inhibitors that feed back into the process that slow it down. One is if there's an excess of ATP. When there's plenty of ATP can act as an inhibitor and feed back into phosphofructokinase and downregulate, slow down the process. And also, when there is an excess of citrate produced during the Krebs cycle, that can also feed back in and act as an inhibitor and downregulate cellular respiration output. So just keep in mind that the process has regulatory function, and these are just a couple of examples of what upregulates and downregulates the cellular respiration process. And there you have it. That's what cellular respiration is all about. This is what keeps life going. This is the energy of life, and understanding it is going to help you down the road, because this involves so many discussions about what happens in our various types of cells that make up our bodies and keeps us alive and living as an organism and all of life. This is how energy is processed and is used to keep life going out there in our living world. So really appreciate that we've got to this level of understanding of such a complex process, and it's just amazing when you see it all work, and you see the it all makes sense, and it balances out, and it's just an amazing process. So appreciate what you're learning, and invest the time now to figure it out, because it's certainly going to help you down the road, because this isn't going to go away. This is what life is all about.